All right, guys, before we get back to this story, let's talk a little bit about DNA so that we're able to relate why I told you that story and the information that we should already know. And if you're hearing it for the first time, that's okay. We will get through this together. So I want to talk about the big picture here. And the big picture is, what is DNA and how does it work? So let's tackle the first question. What is DNA? Well, DNA, you guys know, is the blueprint for life, but what in the world does that mean? Well, what it means is DNA provides the directions on how to build my body's proteins. So we talk about these proteins, but proteins are what give you your traits. Like I have brown hair, you might have blonde hair. So my cells are making different proteins that give me different traits than your cells are building in your body, giving you those different traits. So my DNA provides those directions. Well, the next question is, how do we go from directions into the actual proteins, into building these proteins and giving me my traits? So let's look at this picture. Here is my cell, okay? And we've got our nucleus. There's our nucleus right there. Our DNA is locked inside that nucleus. It cannot leave. Those directions cannot leave. And I've given the analogy in class. When I got hired down at New Richmond, they gave me a manual, a big manual, that had every job in the building and how the directions on how to do that job. So there was a chapter on how to be a teacher. There was a chapter on how to be a secretary, how to be a principal, how to be a lunch lady. But I didn't need all of those directions. I just needed the chapter that was specifically for teachers. So each one of your cells contains that whole manual, that whole set of directions, even though it may only need a small amount of those directions. So the DNA is locked in that nucleus. It can't leave. Well, remember, it's providing the directions for how to build proteins. Well, my proteins aren't made in the nucleus. They're made all the way down here in the ribosomes, in the cytoplasm. So I've got to get the directions from the nucleus out to the ribosomes, my protein builders, but the directions can't leave the nucleus. So there's a couple of steps that have to take place. The first step is the DNA has to get copied, and really just pieces of it, whichever part the cell needs of those directions, needs to be copied into something called RNA. All right, we're going to talk about that in just a second. That RNA then, once that shorthanded set of directions is copied, then it can leave the nucleus, it can go to the ribosome, and from there, the ribosome can read that set of directions, and then it can build the protein that it needs to make. So before we go any further, let's talk about DNA versus RNA. So I think we've known this since 8th grade. You guys had a little unit on DNA, but DNA is a double helix. It's got two sides to it, or a double strand. As we already said, it can't leave the nucleus. And the one thing we haven't really talked about yet, hopefully we know though, is that DNA has 4 bases, and the bases, remember, are found inside the double helix, right here where those lines are. It's what's on the inside, and that's really what makes us all different. But DNA has 4 different bases. It has adenine, which pairs with thymine. It has guanine, which is always going to pair with cytosine. My combinations of A, T, C's, and G's are going to be a little bit different than your combination, which is going to, again, give us different proteins. Now, if we go to RNA, how is that different? Well, one, RNA is a little bit more simple. Instead of deoxyribonucleic acid, which is named after the sugar deoxyribose that makes up DNA, RNA stands for ribonucleic acid, which it has a slightly different sugar. And as you can see, it's only half of the DNA strand. It's a single strand. The second thing, it can leave the nucleus. It's like a little messenger. So it can leave the nucleus and go out to other parts of the cell. And last but not least, the bases. You'll notice if you look down here, All of the bases are the same except for thymine. Instead of thymine, we now have something called uracil. So adenine, instead of pairing with thymine, it now pairs with uracil in RNA, and guanine now pairs with cytosine. So we need to keep those differences in mind as we move forward. Now, going back to that whole idea, we've got to get those directions out. We go from DNA to RNA out into a protein. And we call that process, there we go, a little bit better lighting. We call that process the central dogma. Again, that's how we go from DNA over here all the way to making proteins, which makes us up and makes us all unique. So the first step, we start with DNA over here, and we copy it into RNA right here. That process from copying DNA into RNA is known as transcription. If you break that word down, it's got the word scribe in it. If you've ever heard of scribe, that means to write. So it's literally like rewriting the directions, only in a simpler form. So once we have those directions rewritten into RNA in the process of transcription, a second process has to take place. So remember, our RNA left the nucleus to go to the ribosome. And from there, that RNA gets read by the ribosome, and it pulls in the different ingredients called the amino acids. Amino acids build up proteins. So the ribosome brings those ingredients to the party, and it's going to put them together in the order that the RNA tells it to. And that's called translation. Think about a foreign language like Spanish. When you translate, you're going from one language to another. Well, here we're going from the language of RNA, nucleic acids, to the language of a protein. We're changing it into something completely different. So again, transcription happens first. That's going to change our DNA to RNA. And then translation is going to read our RNA recipe and turn it into a protein. Now that we have that down, we're going to try to relate this back to the story I told.